For years, therapists have been taught to begin treatment by identifying the cause of their client's condition. By understanding the cause, treatment becomes more personalized and effective. Often, counselors would boil the cause into an underlying issue of either nurture or nature. But due to the ever-growing study of epigenetics, we now know that nature and nurture are directly intertwined. Epigenetics is how gene expression changes via modification of the epigenome without altering genetic code. There are two major ways this occurs. The first and further research is histone modification. DNA naturally coils itself around proteins known as histones. During histone modification, epigenetic tags such as methyl will latch onto the genome. Methyl causes it to tighten and silence the gene. If removed, the gene will untighten and become active again, allowing it to be read. Other groups, such as acetyl, will cause the genome to loosen, allowing for it to be read, but deacetylation will cause it to retighten, and suddenly it can't be read again. Methylation and acetylation are not the only forms of histone modification. Currently, there are over 50 known histone modification chemical tags. One example of methylation you see near daily is stress. Stress causes methylation within the hypothalamic pituitary adrenal axis, or HPA. This causes the HPA to respond with a different balance of hormones than in a stress-free environment. This is how your body regulates stress. If the stress is great or continuous, the hormone balance will synthesize anxiety symptoms. The other discovered effect of epigenetics is a change in microRNA. If a gene is activated, a little guy called mRNA reads the genetic information and takes it outside of the nucleus to tell the ribosomes which proteins to synthesize. And here you can see the mRNA telling the ribosome what to do. And the ribosomes are like, yep, I got it. And as you can see here, he is assembling a protein using amino acids, also known as Lego. However, if there are too many mRNA, the cell will have an incorrect balance of proteins. And so, to prevent this, we have microRNA. miRNA will bind to the mRNA to prevent it from reading the gene, thus preventing an overabundance of proteins. There are two types of binding. Partial binding, where the mRNA prevents the mRNA from reading anything so long as it is attached. When the mRNA leaves, the mRNA is free to continue its job. But during complete binding, the miRNA marks the mRNA for destruction. Various enzymes will then come to eat the marked mRNA. Complete binding prevents a surplus of mRNA. The two forms of epigenetic change, histone modification and miRNA, may work together. For example, if there's an increase in miRNA in a gene that leads to the synthesis of histone acetyltransferases, or HATs, which transfer acetyl groups onto genomes, this increase in miRNA will lead to a decrease of HATs, causing deacetylation in other genes. Epigenetic changes can be caused by a variety of things, including great or prolonged stress and diet. But epigenetic tags can also be passed down through generations. Way back in the years of 1944 and 45, Nazi Germany occupied the Netherlands and decided to limit the Dutch people's food. Thus began the Dutch hunger winter. During the climax of the famine, Dutch citizens ate as little as 400 calories a day. This greatly impacted the health of newborn babies during the time. Those who survived portrayed an increased risk of obesity and heart disease. The earlier the baby was in development during the famine, the worse the symptoms. What happened is that the lack of nutrients provided to the babies caused the IGF-2, insulin-like growth factor 2 gene, to demethylate, since the body just didn't have enough methyl groups to methylate the genes it needed to. Even further still, the children of these children were found to have the same epigenetic tags, or lack thereof. So while your genome can be epigenetically altered through your own experiences, they can also be affected by your parents' or even grandparents' experiences. This isn't limited to just physical conditions like obesity, though. This can be seen in the children of Holocaust and 9/11 survivors with PTSD. Just like the grandchildren of the Dutch famine survivors, these children's epigenetic tags are similar to their parents, causing them to experience PTSD and anxiety symptoms despite not having been involved in any traumatic events. Maternal depression is especially detrimental to a child's mental health. Maternal depression encompasses a range of conditions that affect women during pregnancy and up to 1 year postpartum. In particular, prenatal depression, which occurs during pregnancy, may cause additional methylation within a variety of genes of the baby. Some of these result in an increase of cortisol and an increase in the HPA stress response in the newborn. This also increases the chance the baby experiences a mental disorder or other forms of cognitive malfunction. For example, in a study done on nurturing in rats, it was found that mice who had not been properly nurtured by their mothers had much higher methylation in the glucocorticoid receptor in the hippocampus. This caused the rats to showcase more anxiety as their HPA stress response was not responding to stress as it should. Postpartum depression often causes a lack of interest or attachment between mother and baby, causing similar methylation patterns to that of the rats. Epigenetic tags are not only passed down through mothers, though. The epigenetic tags on fathers' genomes are also passed on. For example, fathers with a high miRNA condition will create children with the same high miRNA conditions. In short, epigenetic tags have the potential to cross generations via the process of imprinting. Epigenetic processes occur constantly, but are most active in the first 5 years of life, and so it is incredibly important to create healthy environments for young children. Some things that may cause change include housing, poverty, lack of green space, or excess of pollution, as well as social stigmas. Another issue that causes unwanted change is bullying. Bullying causes methylation in the serotonin transporter gene, which may lead to conditions such as depression. It also causes methylation on the FKBP5 gene, which lessens the response to cognitive behavioral therapy, making mental conditions much more difficult to treat. This is part of why it's so important to stop bullying early, as the effects may make treatment for future mental health issues much more difficult. However, epigenetic tags can change. Just as traumatic events and lack of nutrition can cause unwanted histone modification, positive events, healthy diet, and other changes, such as exercise, have the power to rectify these unwanted changes. Because the first few years of a child's life are so integral to their future mental health, there are a variety of parent-child interactive therapies available. These therapy sessions are preventative in nature and are especially useful for new parents as the sessions cover parenting strategies and often lead to an increased attachment between the family, as well as increasing pro-social behaviors and attention span within the child. However, epigenetic changes are not limited to young children and are affected by daily actions and surroundings. For example, one study's results suggest that a short meditation intervention in trained subjects may rapidly influence the epigenome, causing differences in methylation. As you can see with this sock, meditating. Breathe in, breathe out, buddy. In a study on rats with anxiety, one microRNA was found in larger quantities in rats highly showcasing anxiety than rats that did not. When the mRNA was increased in the low-anxiety rats through artificial means, those rats began to exhibit more anxiety and demonstrated the same behaviors as the naturally anxious rats. This means in the future, microRNA may be able to be used in pharmaceutical treatments for varying mental health disorders, such as antidepressants. MicroRNA may also be used as a biomarker to help diagnose mental health conditions. For example, The miRNA on screen have been identified as biomarkers for schizophrenia. While the epigenome can be changed, the traces of previous epigenetic tags are nearly impossible to erase. In another study, 45-year-old males who had and hadn't experienced childhood abuse had their epigenomes compared. Despite the many years that had passed since childhood, the abused group showcased differences in methylation in comparison to the unabused group. While the epigenome can be altered by positive events, cognitive behavioral therapy, diet, and exercise, and meditation, there is still no true way to perfectly restore an altered epigenome. While medication may change chemical balances or cause epigenetic changes, traces of previous chemical tags will always remain, not just genetically, but also through memory. The memories of traumatic events and bullying are not affected by epigenetic changes, as the only real cure for them is time. As such, it is doubly important to care for mental health before mental illness forms. Whether it be stress, addiction, or loss, if you or your children are suffering from mental illness or are feeling overwhelmed, find a trustworthy friend to talk to, schedule a session with a counselor, or call the number provided, because mental health is important.